Hey everyone, and welcome. Today we're diving into a really cool genetic mystery, something that seemed to break all the rules. We're talking about why some inherited traits just seem to get stuck together, completely defying what scientists expected. So let's kick things off with this puzzle that had early geneticists scratching their heads. They'd see that certain traits, maybe eye color and wing shape in a fly, would show up together in the offspring way more often than they should have. It's almost like they were glued together. This was a huge mystery because it went against the fundamental rules of inheritance they thought they understood. You see, the rule they all knew was something called independent assortment. The idea is that genes for different traits are supposed to get shuffled up and dealt out randomly, kind of like a deck of cards. You'd expect to see all sorts of different combinations, but that's not what was happening. Not at all. And this whole observation of genes sticking together when they should be separate leads us right to a core concept in genetics known as linkage. Okay, so what is linkage? Well, it's pretty much what it sounds like. When genes are physically close to each other on the same chromosome, they tend to travel together as a single package when sperm and egg cells are made. And that is the reason why certain parental traits can pop up in their kids far more often than probability says they should. Now, what's really interesting is that linkage isn't just an all-or-nothing deal. It actually comes in two main flavors. First, you've got complete linkage. This is when genes are so tightly packed together that they are always inherited as a unit. You only get the original parental combinations, period. But then there's incomplete linkage. Here, the genes are still linked, but they can be separated sometimes, which allows for new non-parental combinations to sneak in. So how did we figure all this out? Well, let's go back in time and look at the key experiments that first cracked this genetic mystery wide open. The first big clue actually came from sweet peas, in an experiment run by two scientists, Bateson and Punnett. What they did was cross a pea plant that had blue flowers and long pods with another one that had red flowers and round pods. Simple enough, right? Well, check out these results because this is where it gets wild. Based on independent assortment, they expected a nice, even 25% split for each of the four possible trait combinations. But just look at what they actually got. The parental types, blue and long and red and round, showed up almost 44% of the time, while the new combinations were super rare. The numbers were totally skewed. So, the takeaway here was massive. Something was physically keeping those original parental combinations together. The genes for flower color and pod shape were clearly linked. But because those rare new combos did appear, it wasn't a perfect link. It was a classic case of incomplete linkage. All right, the next huge leap forward came from the lab of T.H. Morgan and his famous work with fruit flies. You know, fruit flies are perfect for genetics because they reproduce so quickly, and Morgan's experiments with them gave us the definitive proof for both types of linkage. In one of his first big crosses, Morgan was looking at eye color and wing shape, and the results were just stunning. Look at this slide. He observed absolutely zero new combinations, zilch. The flies either had red eyes and normal wings or purple eyes and vestigia wings, exactly like their parents. Nothing in between. I mean, this was the smoking gun. It was undeniable proof that some genes are packed so tightly together on a chromosome that they almost never get separated. This was a picture-perfect demonstration of complete linkage in action. But then, in a different cross, Morgan saw a very familiar pattern emerge. You know, just like with the sweet peas, the parental types were way more common than expected. But this time, those new combinations, the red eyes with shrunken wings and the purple eyes with normal wings, they were back, even if they were still pretty rare. Another slam dunk case of incomplete linkage. So this brings us to the really big question, right? If genes are physically stuck together on a chromosome, what on earth is the biological mechanism that can sometimes pry them apart and create these new combinations? The answer, it turns out, isn't about some weird exception to the rules of inheritance. No, the answer lies in an amazing process that literally shuffles the genetic deck while sperm and egg cells are being made. And that process is called crossing over. This is it. This is the beautiful, elegant solution to the whole genetic mystery. It's the physical event that actually breaks the linkage between genes. And you know those rare, non-parental offspring we kept seeing in the experiments? Well, they have a special name. They're called recombinants. They are the direct living proof of this crossing over process. And honestly, they're a huge reason why there's so much genetic diversity out there. So here's how it works. During a special type of cell division called meiosis, the chromosomes you got from your mom pair up with the chromosomes you got from your dad. Their long arms can physically overlap, and when they do, they can actually swap pieces of DNA. So when they finally pull apart, some of those chromosomes are now a brand new patchwork of the originals, carrying combinations of genes that didn't exist before in the parents. It's amazing. And this right here, this is the final brilliant insight from Morgan's work. He figured out that there's a direct connection between where genes are physically located and how often they get recombined. And it's actually pretty simple when you think about it. The farther apart two genes are on a chromosome, the more physical space there is between them. And the more space there is, the higher the chance that a crossing over event will happen somewhere in that gap separating them. So the frequency of seeing these new recombinant types, it's not random at all. It's directly tied to how far apart the genes are. And that leaves us with this incredible revolutionary thought. If we can just count how often linked genes get separated, we can basically measure the distance between them on the chromosome. This single idea was the key that unlocked the door to mapping the location of genes, and it laid the foundation for the entire field of modern genomics. How cool is that?